next presenter is Chris Pryor with Pendrea Pharmaceutical. And I see you, Chris.
Good morning, everybody. It's a pleasure to be selected for this conference. This is Chris Pryor speaking. Pendrea presents a really differentiated opportunity in that we're actually working with a class of compounds called ceramides, which are a major part of the body's natural control mechanism against neoplasia. When the anti-cancer properties of ceramides are depleted, this allows healthy cells to progress into a cancerous condition. At Pendrea, we have a unique, very potent form of a ceramide with potent anti-cancer properties that blocks the high-energy glycolytic pathway of cancer cells. So the target site is unique to cancer cells, but completely safe to healthy cells because the target is just not there. Ceramides are in every cell in our body. So it's safe and it's cytotoxic, which is quite an unusual statement. Furthermore, when we expose cancer cells to ceramides, they become particularly sensitized to chemotherapy, immunotherapy, and this is terribly important in relapse patients who are no longer responding to their treatment. And adding Ciraxa to their treatment regimen allows restoration of this effectiveness to prolong their life, and this really addresses unmet needs and is a very efficient path to approval. The management team consists of our Dr. Vlock, who's a board-certified oncologist, many years of experience and leadership in large pharma, running large cancer clinical trials. My own background is again in leadership roles in large pharma and biotech, and between the two of us, we built 5 companies from scratch, of which 3 have enjoyed lucrative M&As, exits, and 2 IPOs. We have the scientific founders are very heavily involved in the company because, as you will see, we have an underlying technology platform that allows delivery of these ceramides. The ceramide that you see, our unique one with highly potent anti-cancer properties, is a C6 form of the ceramide, and all these molecules are really quite insoluble and need to be delivered intracellularly, so quite a hurdle. So this is why Pendrea is the only one working in this field. The molecule is inserted into the outer membrane of this tennis ball that you see on the left, and that becomes Ciraxa. It's the tennis ball, the particle, with the Ciraxa embedded in the outer membrane. It's stable in humans, it has a long half-life, and its particle size allows it to accumulate in tumors. Once in tumors, as I mentioned, the release of the ceramide inside the cancer cell blocks its glycolytic pathway and triggers programmed cell death. But in addition to that, and this is unusual, we also have an immunotherapeutic, a strong immunotherapeutic component whereby the CD8 cells are reactivated to attack the cancer cells. So you have these dual mechanism of action, these broad outcomes, and essentially the cancer cells are put into a state of checkmate. So we completed a dose escalation in a phase 1 trial, which was really set up as a safety trial. And quite unexpectedly, we saw encouraging signs of efficacy in 5 of 13 patients in the dose range of 81 milligrams, where we saw the activity. Ciraxa has shown remarkable regression in the Nivo model in cancer, liver cancer. And on the right-hand side, you can see the remarkable sensitization to chemotherapeutics. We're looking to raise $30 million to get to the end of year 2 to complete phase 2A in a basket of solid tumors and we will choose the one that has the highest overall response rate. Similarly for AML at the end of year 2. So in summary, why this program is de-risked. Our monotherapy results that we got for efficacy is expected to be greatly enhanced in the presence of chemo. We have 4 shots on goal with indications, AML adding the 5th, low bar for approval in relapsed patients, and excellent safety. So thank you for listening, and we'd be pleased to answer any of your questions, and please follow up with email. Thank you.